Okay, good evening, everyone, and we are starting now the afternoon session on mass spectrometry, and we have the first guest speaker, which is going to be online. Annemieke, are you there?
Yes, I'm there.
Okay, hello, Annemieke.
Hello, Kostas.
And Annemieke Heijboer is a full professor of clinical chemistry in the University of Amsterdam, and also chairing the laboratory of endocrinology, and is also a full member of the IFCC Committee on Bone Metabolism. And we thank you very much for your participation in this symposium. And her speech is going to be by endocrine testing using mass spectrometry. So, Annemieke, the microphone is yours.
Yeah, thank you, Kostas. I would like to start with thanking the organizing committee and especially Kostas for inviting me to give this talk. I'm indeed working in the Amsterdam UMC, which is a university medical center, and where more than 16,000 people are working on patient care, research, and education. And besides working in Amsterdam UMC, I do not have any disclosures for this talk. I'm heading the endocrine laboratory, where we do the quantification of hormones in all kinds of bodily fluids and also some tissues. And we work there on diagnostics, so we quantify the hormones for the patient care, but also for research studies, and we do research and development ourselves. In our laboratory, we have immunoassay platforms from different manufacturers. We perform the neonatal screening for part of the Netherlands. And we do have some LC-MS systems to quantify the hormones. And that's, of course, what I'm going to talk about in the coming minutes. And especially, why should you use mass spectrometry for endocrine testing? Well, I think the most important things are the specificity, the independence of matrix or specific patient samples, and profiling. And I'm going to give some examples to show why this is important. And I will start with testosterone in samples from neonates. Well, regularly we get the request to measure testosterone in these samples, and that's often when a baby is born and it's not clear whether it is a boy or a girl, because the gonads are somewhere in between. And besides the genetics, the pediatricians often want to know the testosterone concentration because it will help in understanding whether there's undervirilization or overvirilization, and because it will help them to take the next steps. When we are going to measure the testosterone in these samples, we can, of course, do this with an immunoassay or with LC-MS. And we know already that testosterone immunoassays suffer from cross-reactivity with similar steroid hormones. And a few years ago, there were some second-generation immunoassays on the market for testosterone with less, who suffer less from cross-reactivity. So we thought, let's try and see whether they also are better in measuring testosterone in the samples from neonates, because the neonates, their adrenals are not well developed yet. So some of the enzymes are not working, and they have optimally, so they have high concentrations of several steroids. And what you do see is that, of course, in the first generation assays, but even in the second generation immunoassays, you do see a lot of cross-reactivity. And what you can see in this figure or in this graph is that especially in the first days, week of life, the immunoassay for testosterone measures much higher, falsely high testosterone concentration. So this can be up to more than 10 nanomoles per liter higher than when you measure it with LC10-MMS. So this cross-reactivity leads to a misinterpretation, problems in understanding whether this child has a high testosterone or not. So I think this is already a good example of the cross-reactivity you do not want to have to interpret for a right interpretation of the pediatrician. My next example is also about testosterone, but then in men. Until a few years ago, I always thought testosterone in men with high concentrations, it doesn't really matter whether you measure it with an immunoassay or with LC10-MMS, because I thought that these levels are so high when you have a little bit of cross-reactivity, whether a testosterone is 20 nanomoles per liter or 21 or 22, that doesn't matter for the interpretation. But I think this is a very nice example that it's also very important in high concentrations to measure very specific. And when you're talking about testosterone in men, and when you Google this on the internet, you see a lot of figures like this, where they show that testosterone levels decline with age in men. And also this nice review in the Lancet also concludes that testosterone concentrations decrease with age in men. And almost everyone thinks, all physicians in our hospital, for instance, have in mind that this is the physiology. All this information, however, is based on a lot of studies that all measure testosterone with immunoassays. And when you do measure the testosterone in a man of different ages with LCMS, you do not see a decline in the testosterone concentration. So this is one very early example, already 10 years old. But here and afterwards, many more studies repeated this. The testosterone in men, the average testosterone is not lowering, it stays the same over age. And this is the difference is caused by a lowering in DHEAS, because that adrenal hormone that lowers during when aging, and it cross-reacts in the immunoassay. So at a younger age, you have more cross-reactivity, so the testosterone seems higher, but it is the DHEAS that lowers, that looks, and thereby it looks like the testosterone is lowering. So this is also an example of cross-reactivity, even in high concentrations of men, which led to a wrong interpretation of research data. Then I would like to go to the matrix independence of the LC10-MMS. So with the matrix, we mean everything that's in the sample, which is not the analyte of interest. And so it can be, if you want to measure in EDTA plasma or heparin plasma, sometimes that's a problem in immunoassay. So the kit insert often says you can measure in serum, but not in EDTA plasma, or vice versa. But it's also, the matrix is also different in different patient groups. So for instance, pregnant women have high binding proteins. Patients in the IC unit have low albumin. Patients with kidney failure have high ureum. So this can all alter the serum matrix, and that can lead to differences in the measurement in an immunoassay. And I will give you an example. I will give you a few examples. Here you see, for instance, the free T4, the thyroid hormone measurement. So on the x-axis, you see it measured with LCMS, and this actually is the reference method. On the y-axis, you see the free T4 measured with an immunoassay. And you see the method comparison for both healthy controls and for patients on dialysis. And you see that there's a difference in how the immunoassay measures the free T4. So it's falsely low in patients on hemodialysis. This is not only for this particular immunoassay. All manufacturers of immunoassays show these problems. So here you see another immunoassay, and it's not only for patients on dialysis, it is in samples of patients, of all kinds of patients. So here you see an example in pregnant women, where you can also see that they are measured falsely high compared to healthy controls. And it's not only about free T4, it is also you see this problem also in other immunoassays for other hormones. So here you see, for instance, the testosterone, which is different in pregnant women from women using oral contraceptives or not using oral contraceptives. Here you see it for cortisol. In this case, the difference between the hemodialysis patients and healthy controls. And here you see again an example for testosterone, but again with another immunoassay. So this shows that immunoassays have a real problem with their matrix for hormone measurements. And often in the kit insert, you see a method comparison healthy controls. But what we do measure in clinical laboratories is mostly not healthy controls, but we do measure patient samples. So I think this is another reason that it's an advantage to measure with LC10-MMS. Then my last example is a case with hypoglycemia. It's about a 72-year-old man. He's found by his roommate with decreased consciousness. His glucose is 0.5 millimoles per liter. And the physicians also see a small subcutaneous hematomas. So therefore, they have a suspicion of hypoglycemia factitia by administration of exogenous insulin. The patient himself, he denied he had injected insulin, and he denied a suicidal attempt. Of course, the physicians were going to do some measurements. They did not measure sulfonylurea, but they measured a high insulin and a low C-peptide, which is of course still suggestive of exogenous insulin administration. But it's a difficult conversation with the patient as he keeps on denying. So it helps the physician. It strengthens the suspicion of the physician, and it also facilitates the discussion with the patient if you can say which kind of insulin analogues were found in the serum. So we have developed an LCMS method in the past where we can measure all insulin analogues, and we could measure ephedra and lentes in his serum. And that helped the physician to start this conversation with the patient. And in the end, it turned out that his roommate has ephedra and lentes because of diabetes. So it was clear that he was really injected with these insulin analogues. So this is an example, I think, how LCMS can help you. And also an example of profiling, because if you measure with an immunoassay, you only measure insulin, and often the insulin analogues cross-react in the insulin assay, but not all analogues cross-react in all immunoassays. So it's more difficult to interpret. But when you measure with LC-MS, you can really quantify all different insulin analogues, and that improves, in this case, the interpretation and the conclusion of the physician. So is LC-MS then the ideal method, and should we always measure everything with LC-MS? Well, there are many method comparisons in literature, and this is just one example where you can see that LC-MS methods can compare really nice. But there are also method comparisons in the literature which shows that it's not always ideal. So it's very important to develop the method well, to validate it, because otherwise you can, in a not well developed method, it doesn't have advantages anymore. And I want to show you here what can be the advantage and how important it is to validate the method. So this is an example of testosterone in the female concentration range. A lot of samples were measured with several immunoassays from different manufacturers and with several LC-10-MS methods from different hospitals. And when you look at the inter-method variation of all the samples, the inter-method between all the immunoassays, you see a much higher variation than when you look at the inter-method variation of the LC-MS methods. But within the LC-MS methods, you also see a difference between published and unpublished methods. And I think in general, the published methods are much more thoroughly validated than the unpublished. I'm not saying that unpublished methods are not well validated, but you do at least see a difference. And I think this underlines that the amount of validation and attention that's paid to the quality of the LC-MS method can lead to an improvement in the inter-method variation. So in conclusion, I think there are some advantages of well-developed LC-MS methods in hormone testing, that specificity, that they're not matrix or patient dependent, and that you can do profiling. And this together leads to a more reliable conclusions in diagnostics and research. And if you are interested in specifically in the quality of hormone methods, we wrote a mini review in JCM a few years ago, which gives even more examples. And also on our LinkedIn page, we also regularly post topics about the quality of hormone testing. And then I would like to end this presentation with thanking you for your attention, and with thanking all the people in the lab who did all the work. And I'm happy to answer any questions.
Thank you, Annemieke. Are there any questions for the speaker? No? Okay. No questions. It was very clear, and thank you very much for this beautiful presentation, and also that that was the first speech that also pointing the importance of profiling the natural measurement analyte that we measure, and also the artificial that or the pharmaceutical that are already in our organism as therapeutic agents. Okay. Thank you very much, Annemieke. Hope to see you soon.
Yeah. Bye.
Bye.
Bye. And now we continue on the second presentation of the symposium with Professor George Theodoridis. He's a professor of analytical chemistry at the Aristotelian University of Salonika in biomics laboratory, and he will talk about the identification of biomarkers using mass spectrometry-based metabolomics assays. George, you have the podium.
Many thanks. It's a fantastic symposium. I'm humbled to be here. Congratulations to the organizers. So This is the group you see on the photograph, and this is the lab. It's a bioanalytical lab. A lot of mass spec and LCMS, and actually, I'm not a mass spectrometrist, I'm a chromatographer. So, right, okay. Acknowledgements to the group, a number of people that did all the work, enthusiastic, very talented guys, and also I would like especially to thank the industrial collaborators from this day. I will speak only about results with Waters Corporation, Lee Gettings, and Ayasa, and the others. We have funding, I have to acknowledge, from European grants, EU Horizon, we coordinate the first two, and we participate in others, non-communicable disease, exposomics, things like that. Now we are starting a new grant in the next weeks. It's a major initiative on patient-centric sampling. I'm not allowed to give any information on this. And also we have some EU fora from EFSA and PRIMA grants and some national grants as well. And so, after the talk of yesterday of Brian Rappold from LabCorp, I put 5 slides more, or maybe 7, so I will rush. I would like to be also provocative. Why do we discuss analyte identification for metabolomics? It is a big problem. So in 20 years now, the last 20 years, we have now a huge number of publications in metabolomics. The routine is the same, and metabolite identification remains a bottleneck. It is time-consuming, and it is error-prone. So this is the routine. This is a workflow. We do full scan. We have some terabytes of 3 or 4 dimensions, 3 or 4 dimensional data. We collapse this dimensionality to generate a peak table, an Excel spreadsheet or something, which we do statistical analysis with multivariate analysis. Here, every dot is a different sample. When these are, the populations are different, then we try to find markers and make very beautiful visualization, and then go to different databases to find what it is and build biochemical hypotheses, disease mechanism, and biomarkers. The problem is that all this is based only on one thing, HRMS data, just a number. And there is no verification, and this is in direct contrast to the guidelines, and there are plenty of them. So Lloyd Sumner, 2007, proposed 4 identity levels. Szymanski, Emma, some years later, 5 identity levels, and then Sumner came back again with 5 identity levels. There are direct guidelines from EU, decision 2002-657 from 2002, 4 to 5 ID points. Now it's a regulation 2021. Again, 5 identity points that are measured by separation, 1 point, LCMS ion, 1 point, HRMS ion, 1.5 points. You sum up, you are 5, you are safe, more or less. And all this have to go on a matrix match standard, on a matrix 45 standard with a tolerance of in LC 0.1 minute, and be done in the same lab with a specialist that has been doing this for years. And the literature disregards this. So there are a number of published papers. I am editor of Journal of Chromatography B. I reject 80% of the papers that come. This is my statistics. Nevertheless, of course, a number are published, and I will show some results. Now, what we identified is 2 types of errors, the poor biological plausibility and identities that do not comply with chromatography. We wrote a perspective in 2021. We wrote another perspective last year with the editor of metabolomics, with the editor of analytical chemistry, and other guys. The Smiths they think, what difference does it make? It makes none. Yes, it makes none. CRAP is published. One example now, In the previous perspective, we had a critique we do not name. So I am naming now paper, adjuvant-induced arthritis rat model, impact factor 4.5, 1,000 exogenous substances identified and given names, like beclomethasone, a typical, synthetic compound, and so many other such markers, biomarkers of disease, identified. Then here, another paper, Lewis rat model experimental autoimmune neuritis, impact factor 8.3, and they find mitoxantrone and methylprednisolone. These are both synthetic. And they characterize as down-regulated molecules. And then they identify more, names like a mile long. This you cannot identify. Nobody can identify these things. Top of the top, serum metabolic profiling and metabolome genome-wide association study in chicken. Chickens from the farm identified 7,000 exogenous substances. So there are so many bells that are ringing, nobody's listening. They identify 75 INN, that is internationally non-proprietary. These are typically pharmaceuticals, and these are some examples in red. You see drug, herbicide, synthetic, all this, and many more in the blood of the chicken, like they consume the whole pharmacy. Second case is that they disregard physical chemistry basic laws. So what we see here is an elution order that is absolutely impossible. Apolar, polar, apolar, polar, apolar, polar, and so on. No way. So there are possibilities to do, and you need to corroborate, and of course, all this is LCMS data, and the LC is there, the LC data is there, and nobody is using this. So one is this, use the LC data. And so I would say that metabolite identification is not for everybody, and we need to go back to the basics to avoid such problems. Now, enough with this, and I will come to the other part of the meta-ID, what we do in labs, in different labs, to identify biomarkers, not necessarily with untargeted analysis. So in our lab, we develop LCMS and GCMS methods for small molecules, exogenous and endogenous. We contribute to biomarker discovery with other colleagues, and we apply untargeted metabolomics and lipidomics in various applications, for instance, multiple sclerosis. And recently, we are very active in investing a lot in microsampling and alternative sampling, like patient-centered sampling, including bridging studies between blood or whole blood and dry blood spots and so forth. So our portfolio has roughly 50 fit-for-purpose methods. Some are very well, very highly applied, more than 3,000 samples, high TRL. We know them very well. So some examples, short-chain fatty acid by GC in feces, mainly on inflammation or cancer. So, acetic acid, propionic, butyric acid, and so forth. Amino acids in blood, urine, and dry urine spots, fatty acids, 55 organic acids in urine. We are now moving into steroid panels. And we have other panels like carnitine, ceramides, vitamins, biogenic amines, and so forth. One we are very keen on is of this that is now about to be published, aromatic amino acids and their sulfated structures. So we synthesized all these blue molecules here, around 12, so 10 and 2 internal standards. And so we have now 39 metabolites detected in urine. We validated this, and we're about to submit. And we are now validating also in blood and saliva. And the aim is metabolic syndrome cohorts and linking metabolomics with gut microbiome to study the gut-brain axis. Another study we invested a lot is the physical exercise. We have realized maybe 6 PhD studies. In this case, we have we coordinate this Marie Curie network, 12 PhD students. The application we do in Thessaloniki is intensity exercise. So there we collect the blood at the spot, and this is very important. You need to take the snapshot of biochemistry at the end of the physical exercise, not 3 hours later when people recuperate. So we selected to do this with dry blood spots. And 3 devices and lipidomics analysis and different metabolomics analysis with different modes. Just one example here, we see the 3 devices and how some metabolites are looking in capitainer, in mitra, in Wattman, the normal Wattman card, and the native plasma. And so we see that things are different depending on the different type of the dry blood card that you use. I will not go in much detail on this. I want to proceed with Alzheimer. Here we have a cohort of several different cohorts actually, but just an example here, we analyzed with waters semi-quantitative screening of lipids, untargeted metabolomics, and PFAS. 3 methods. So this is how it looks, the clinical data. And there are 3 more screens with different clinical data. And I'll go through this. Just the result, okay, we do separate in lipidomics the Alzheimer from the controls, whereas Alzheimer from MCI or control from MCI do not separate so well. The new one that is, we are very happy is the detection of PFAS. So we see this is the first time PFAS is detected in this type of cohort. We see some PFAS detected. And then we moved on to a new technology that is the cyclic ion mobility that can separate isomers. And here we see how the different isoforms of this compound are separated in the ion mobility. And this is something that cannot be achieved by mass spectrometry alone or by chromatography alone. So I would like to conclude with this study, the correlation of severity of artery disease, coronary artery disease patients' metabolic profile. We started with 1,500 participants, finally 1,200. We identified by machine learning in the literature ceramides, carnitines, amino acids, fatty acids, and some proteins as, along with the typical biochemical tests as potential biomarkers. And we would like to predict the coronary angiography, the digital angiography results, and the syntax score, which is the golden standard for this disease. Some demographics, so classical biomarkers, cholesterol, troponin, imaging technologies, and our new biomarkers. So we developed the method for ceramides, we developed a new method for carnitines, and some statistics here, how these are differentiating between STEMI and non-STEMI groups or other groups of clinical severity. And then again, syntax score versus our data. This is the acyl carnitine data, how it looks. Some are found very important, C5, C10, C16, C18, and so forth. And then actually the correlation of C4 and C18 score underscore colon 2 is very highly correlated to syntax score and can predict it. And in the end, these are the fatty acids. In the end, we have this big list, and I will skip this. We use machine learning to put all this together, 5 sphingolipids, 7 glycerophospholipids, 3 alkyl galactine and other molecules and markers as a 17 biomarker panel that can predict the severity. And this is now launched in the market, to be launched in the market for, and give an example of the utility of LCMS. Thank you.
Thank you very much for your presentation. It was very interesting. And have we questions in the audience? Georgios, how is you think a laboratory that has mass spectrometry on his equipment to apply these techniques?
Can you repeat the question?
How easy is it for a laboratory that already uses mass spectrometry to be launched in biomarkers like this, the untargeted lipidomics?
Lipidomics is not difficult. I mean, it can be demanding. So if people are trained, they can do. It's better understood, and I think it is more advanced. Anne presented this earlier. Than untargeted metabolomics. Untargeted metabolomics is analytically dependent and will come very slow. Targeted analysis, now there is a fusion between clinical chemistry and targeted analysis because it's the same thing. We measure amino acids or organic acids, or things like that. Okay. So that is the first step. Go to what you know well and step by step move forward. This is what I would say.
Thank you very much.
Okay.
Thank you.
Thank you very much. Now, we move to the next speaker and the final speaker of this session, which is Giovanny Rodríguez-Blanco, which is a chief scientist, senior scientist in the University of Graz in the clinical chemistry department. And he's going to speak about the expanding capabilities of LC-MS in newborn screening. It's another interesting area of application of this technique.
Okay. Let me see, how does it work? Okay, sorry. Good afternoon, everyone. First of all, I would like to thank the organizers for giving me the opportunity to show the data and all the progress that we've been doing and all the new data that we are collecting in Graz. For the ones who maybe don't know, I'm coming from Graz in the south of Austria. So, Graz is here, Vienna is here, and basically we are the second largest city in Austria. And basically I work at the LKH Graz, is the university hospital of the region, and I'm also affiliated to the MedUni Graz, which is basically one of the biggest medical universities in Austria as well. So within the LKH, I belong to the KIMSIL, which is this long name. The KIMSIL is headed by Professor Herman, who is sitting in the audience today. I'm also a scientist, and by training, I've spent more than 10 years dealing with high-resolution mass spectrometers. So we saw a similar picture earlier today, but I like this one because it shows how the cell, in general, can be explained using the systems biology approach. As a mass spectrometrist, I'm not interested in this bit, although we can identify some RNA modifications with the mass spec. But I've done a lot of proteomics, and obviously small molecules is the thing that I'm pushing forward. We heard today also the beauty of the lipidomics and how quickly it's moving into the clinic. As I said, I belong to the KIMSIL, and within the KIMSIL we have two labs. The main lab is the routine lab, where we have all the Cobas and Siemens instruments for routine work, and the mass spec lab, which is the one I'm in charge of, is the special analytics. And the special analytics is divided into basically the TDM, the therapeutic drug monitoring lab, where we run on a daily basis immunosuppressant antibiotics, antiepileptics, metanephrines, and neuroleptics. So this is a pretty robust and established lab. We work pretty much 7 days a week. And I have to say that most of these methods, 95% of them are already with kids, so they are IVDR regulated, they are good to go, and they are pretty solid. The other part of the lab, which is the Stoffwechsel, or in German, metabolism in English, is basically the one I'm the head, let's say the head, is where we do all the metabolic analysis for the kinder clinic. There are about 20 different methods that we run in the Stoffwechsel area, but I'm just highlighting these 4 because these 4 are the ones that keep me busy the entire week. So we have methods for amino acids, organic acids in urine, acyl carnitines, and bile acids. The difference with TDM is that only amino acids is coming with a kit. For the rest, we have in-house developed test. And obviously with the new regulations with IVDR and also with my experience in the omics, I want to improve and I want to expand the capabilities of this method to improve the diagnosis. So I'm going to start giving you an update of the methods that we have in the lab. So when I joined the lab 3 years ago, we had this amino acid analyzer. It's a beautiful machine. It works really nice. It's really robust when it works, but it uses 250 microliter of sample, and then we use the instrument for about 3 hours. The separation is really good. It's not mass spec, but you can see base separation for up to 22, 23 amino acids. What happened early this year was that we run our standard, we see a nice separation, and this is the peak here for arginine. When we run a patient from a kit, you see that the peak nearly at the end of the chromatogram was pretty much gone. So we flagged this sample, we had to go to the clinicians, and turns out this was a false positive. Then we decided to use our kit, our amino acids. We are collaborating with Chrome Systems because they have this established kit. So we moved from 22, 23 amino acids with the ion chromatography to 48 amino acids in only 20 minutes, and we only use 25 microliter of sample. So this is the chromatogram. This is nothing informative here, but one of the things that we like the most out of this method is that it's fully integrated with our laboratory information system. So we are speeding up things, we're improving the diagnosis, and we are getting better data. This is a bit of the data from the validation. Obviously, we have our 3 level calibrators. We have the pathological and normal controls, and we have the 3 levels of QC. For this comparison, I also included 2 different external QCs from a different company. And what you can see here in this PCA plot is that these are the calibrators. They cluster together with all the technical replicates we have. These are the external QCs from the kit. Also, they cluster nicely together. And interestingly, the external QCs from the different company cluster far away from the others. So it shows that the method is quite versatile and is quite unique. This is another way of representing the data. What we realized is that, for example, for the 1-methylhistidine and for the 3-methylhistidine, there was a mismatch with Chrome System and the other company, and this is a matter of investigation. But in general, the method is quite robust. We are happy with the method. The method is good to go. However, the kits are not perfect. And sometimes the company is having problems in manufacturing both the column and the QCs, and this is something that we really need to work. So part of my job is to be always on top of the data, always on top of what we are getting to basically get reliable data. That's the ultimate goal. Okay, the next method I want to basically talk about is the organic acids. The organic acids is probably the method that we use the most in the lab, is the determination of organic acids in urine. For that, we use a gold standard method using GCMS. Basically, this is in summary what we do for the sample prep. The ones who are doing GCMS know that the sample prep is not as easy as we would like it to be. And this is the chromatogram. The method is solid, is robust. We also participate in the external quality control from the ERNDIM, and in general, we do pretty well. However, when I joined 3 years ago the lab, what I found was that the clinicians were interpreting these spectra by hand. And for me, it was a bit strange because obviously we can now export all the data in a CSV, TXT, you name it. And for me, it was strange that the clinicians were still getting printouts out of this data to do the clinical interpretation, okay? And then I started digging in literature, and then I came across these 2 papers. One is from 2019 from this group in the Netherlands, and this one is recent from last year in CCLM from a Czech group. And what they did was develop a method, one for the QTOF and the other one for the SciEx 6500, where they increase the number of organic acids that they can be quantified, but they did it by LCMS. And the most important thing is that they increase the number of analytes that can be profiled. And this is basically the inclusion of acyl glycines and carnitines in urine. So this method in particular includes 300 transitions, and they did a fantastic job validating all this. So considering that I like LCMS, I'm not saying that I dislike GC, but sometimes GC is more tricky, and then we have a lot of samples to run. What I did was trying to implement this method and see what we get. And this is the work that one of my master's students is doing. So we took these two papers as a reference. We took the internal standards and the standards that we have, and we started trying to quantify about 25 organic acids in urine. So this is a short method. The beauty of this method is to just dilute your urine and then you shoot it. It uses polarity switching. So this means we acquire the organic acids in negative polarity, but the carnitines and the acyl glycines in positive polarity. This is the TAC, and this is the peaks that you see. In general, this method is about 20 minutes. So this means in about 20 minutes, excluding data processing, you can get entire profiling for the organic acids. One thing I forgot to say, and this is interesting, is these guys came up with this network representation of the newborn screening. So for this, they export the data from your mass spec. They use Cytoscape, which is a network software, to build these kind of plots. And depending on the intensities and the concentration, you can visually see if the carnitines, for example, in these patients are upregulated in your, or are increased in the sample. So what we got was not quite what. So the first thing we did with the standards that we have is that we check linearity. In general, the linearity is more or less in accordance to what the guys in the Czech Republic published. So this is quite good. But however, when we check the inter-day accuracy, you see that there are some red flags. I went back to the papers, I went back to the literature, and the guys from the Netherlands reported a list of metabolites, the organic acid in their method, that they are not quite stable. And for example, glycolate is one of them. You see that this percentage, the accuracy is far away from what we want. So now, after this conference, what we need to do and to finish Anna's thesis is just to go back, revise what everything needs to be revised, and to basically validate if what is published in literature is true, and if we could consider in the near future, after the entire evaluation, if we could implement this method into the clinic. Okay, just the last two methods, carnitines. I've heard and I've seen today that a lot of labs measure acyl carnitines in serum and urine. Usually, if you go to literature, if you go to Cambridge Isotopes or even Chrome Systems, you have standards and internal standards for about 15 acyl carnitines. But this paper was published a couple of years ago. It's a really nice, it gives a really nice overview about acyl carnitines. And what is known so far is that the carnitines are really diverse, and in theory, we could identify more than 1,000 carnitines in a sample. And then obviously, apart from the known ones that we have in the lab, there are saturated, unbranched, monocarboxyl, and so on and so forth. And there are some novel or newly reported carnitines that can be detected with the mass spec, and this is the ones I'm interested in. As I said, I want to expand in a targeted way the number of species that we can not only identify but quantify. One of the good things about the carnitines is the fragmentation pattern. So in this paper, they basically explain this in a very simple way. For the carnitines, you always have this peak at 85 and this peak at 60. This is unique for the carnitines, and the rest information comes from the carbon chain. So for example, in the C18, you have the parent ion here, and then the 85 and the 60. And when this carnitine, the acetyl cysteine conjugated carnitine, you see a more complex MS2 spectra here, but you see still the same 85 and 60 peaks. So if I go back to literature, for example, if I want to develop an MRM, I take as a reference this paper published in JLR in 2015. And as you can see here, this is the list of transitions. All of them include the 85 product ion, and they claim that they can separate the isomers. You see these carnitines have the same precursor ion and the same product ion, but then they can be separated chromatographically. This is more or less what I found when I joined the lab. However, when I tried to run the samples using my methods, using what I had at the time, what I found was this: I'm not able to separate the isomers. Therefore, the identification is tricky and the diagnosis is not accurate. So what I did was I started developing the method from scratch. This method uses a butanolic HCl as a derivatization, but there are other methods, probably you've seen them already, that without derivatization you can profile carnitines. But for now, by changing the chromatography only, we can see, this is data that we produced recently, you can see it's not quite base separation, but still we can separate the isomers for these peaks. So I hope that in the next meetings or in the next few months, we should be able to get the method that is up and running, and then we can use it for clinical purposes. Okay, for the last method, I want to deviate a little bit. As I said in the beginning, I'm a scientist by training, and what I'm trying to do ultimately is try to incorporate the omics and the untargeted profiling that I used to do in the past into the clinical work. And the bile acids is the perfect example of what is happening. I've seen today methods for the classic primary, secondary, and glycine and taurine conjugated bile acids. Usually, Annie mentioned this method today is about 15 bile acids that can be quantified in serum. But what is nice and what is happening is the following. So first, I'm going to mention this paper. This paper is a Nature paper from Peter Dorstein's lab. It was a Nature paper where they, for the first time, described a novel set of bile acids, which are the amino acid conjugated bile acids. In this case, they found them in stool, but it's likely that they are also present in serum, and because the bile acids are no longer considered the surfactants for lipid absorption, but they are considered lipid and signal molecules, they have a momentum for biology and for translational research. The second paper I want to highlight is this one. It's a Comms Biology paper published early this year when they show the diversity and the composition of the bile acids in different sample types and in different organisms like human rats and mice. As I said, what everybody wants to profile with a clinical applicability is primary, secondary, and glycine and taurine conjugated bile acids. However, as you can see here, the sulfated and glucurinated bile acids are completely ignored, and they may represent or they may have a clinical value. For this, I'm using a different approach. So as I said, we want to do it transversal or translational kind of thing. So we have an animal model in the lab, we have human samples that we can benefit from, and now we are taking advantage, of the Orbitrap, our brand new team stuff, and also the SciEx, just to get the most out of the bile acids. For that, obviously, the sample prep is a bit more complex, the data processing is even more complex, so we take advantage of software like MSDial, MCMine, and also spectral libraries and platforms to process data like GMPS, if you are not familiar with them. And this is, for example, what we are trying to do. Obviously, we do a lot of things from literature, and then we rely on spectral libraries. But for example, if we check this peak that we know the mass from here, we identify this peak. So what my PhD student is doing is try to annotate the spectra manually to initially confirm the identity of this sulfated glycocholic acid. We're doing it for multiple peaks. This is basically a lot of manual work that he's doing, but he's doing pretty well. And this is interesting because it is taking us to a further development. So in the lab, we have an animal model of high-fat diet. These are rats. Excuse me, I totally forgot the type of rats that we have for this, but these are rats that had high-fat diet, high-sugar diet, or the combination of both. And then we found this feature with this molecular weight and this fragmentation pattern in serum. And for the first time, even though this paper is not published yet, we found that the alanine conjugated cholic acid was upregulated and it was present in this serum sample. Obviously, this is in silico. We know that we need to get the proper standard to verify both retention time and fragmentation pattern, but this is the way that we are trying to do. Right now, we order standards for the sulfated and for the glucurinated, and then we are trying to collaborate with some synthetic chemists just to help us to synthesize these because they are still not commercially available. All right, so I'm going to wrap up now. Thanks very much for your attention. I hope I convinced you that we are trying to expand the portfolio of analytes that we can identify and quantify because the implementation of these methods in the clinic is just a reality. And finally, I would like to thank my team and the collaborators involved and all of you for your attention. And I'm happy to take any questions.
Thank you, Giovanni. Are there any questions? If no, thank you again, and for your participation and the beautiful information that you showed us. And I think now that everybody is ready for a coffee. Yeah.